So today we're actually kind of starting down the path of thinking about questions that people have brought to us. So today's talk, Dr. Manching will actually be talking to try to help answer the question that we've gotten from a few people that we work with, thinking about if you had the ability to take images in your field season, what would be some suggestions on making sure that you're doing it in a way that you can use those things downstream for image processing and that kind of thing? So today, Dr. Manching is hopefully going to be answering some of those questions. And if you all have questions, we'll do the same as we've done before, just doing question answer session at the end. So Dr. Manching is joining us as a part of the OnRamp, the BI OnRamp team. And she got her PhD working at the University of Delaware in a plant breeding program with Dr. Randy Weiser with working on maize. So you'll see probably a lot of the stuff that she's working with now is quite outside of maize, but hopefully you'll find it interesting. So without that, Dr. Manching, go ahead.
Thank you for that intro. I'm gonna see if I can get my screen to share. Good. Can you see it, Amanda?
We can. Looks good.
Awesome. All right. So I'd like to kind of start off by really saying thanks to Amanda for kind of bringing up this idea. And I think it's a really good sort of interlude between some of the talks that have occurred so far within this seminar. And so I'm going to take a slightly different approach to my presentation and kind of format in a way that provides a little bit of background, but takes more of a workshop type approach towards the end. And so this will be mainly focused on image-based phenotyping and some of the do's and don'ts or components of obtaining high quality images. And so we'll start off with kind of a quick introduction to the BI OnRamp initiative. I know that we've worked directly with a lot of people here, but I know there are some new faces. And so we'll kind of just provide a little bit of background about what we do. Provide a quick intro to what image-based phenotyping is. And then go into what the components are of a successful imaging project. Talk about image acquisition, specifically using Fieldbook. And then I've created a little bit of a tutorial on how to take images with Fieldbook, importing and exporting, and then kind of finish off with some next steps and final thoughts. So Breeding Insight OnRamp is a USDA ARS initiative that was established to jumpstart the process of integrating advanced breeding technologies into breeding programs. Amanda mentioned that I have a strong background in maize, and a lot of the resources that are available in some of those big name crops haven't really reached the same level in a lot of the specialty crops or crops with very complex genomics or lack of resources. And so we are a group of 3 people. Amanda is the director. My colleague, Dr. Keo Korak, has worked specifically with the sugarcane breeding program. And then I've done a lot of work with citrus. And so you may find that some of the content here is from citrus itself. And those are just direct examples that I've obtained over the last year and a half or so. What's really unique about this group is that we have a very diverse background. We span computational biology, software development, as well as data mining techniques and traditional like plant breeding, genome to phenome type statistical approaches. But we all have a strong background in plant breeding. And so we really serve to kind of bridge this gap between traditional field breeding, or a lot of what you see on the ground, and what it really takes to move towards advanced breeding strategies, which are sometimes encompassed by big data or complex mathematical modeling or statistical modeling. So we really help try to translate what those are and provide a way that breeders can actually access those technologies. And so we do this by trying to build a USDA transdisciplinary breeding team that's really focused on using approaches for data-informed breeding, providing data analytics, and advanced phenotyping strategies so that we can really make these technologies accessible to our breeders. So plant breeding and advanced technologies. So part of what we focus on for OnRamp is really establishing the foundations that are really important to be able to use some of the advanced breeding technologies. And these can encompass a wide variety of different types of strategies and technologies, but really what's at the core of being successful using any of those approaches is really defined by how strong your foundation is. So we spend quite a bit of time, and you can see by this arrow on the left, that darker green color just indicates that we spend quite a bit more time on this than we actually do using the technologies itself, because this is really what's going to be the important component of using that. And so we try to accomplish building that foundation by working with our breeders to identify or help establish clear objectives within their programs, evaluating and developing effective strategies for obtaining data, also managing the data that may exist on a computer, or actually how they manage the data associated with what's actually in the field. We try to provide open communication about challenges and goals and interests, as well as try to provide a source of guidance in our areas of expertise. But we really try to be receptive students in the areas that are not. So a lot of those commodity-specific components that we may not be familiar with, but are still based on the broader framework of plant breeding. And so if we can really focus on establishing this foundation, then we provide the platform to actually use some of the advanced breeding technologies, such as high throughput phenotyping, phenomic selection, genomic selection, or any of the other future omic selection prediction concepts that are popping up throughout the literature. In addition to that, we really focus on what the main goal of plant breeding is, and it's to increase the selection response. And so I'm sure a lot of you are familiar with the breeder's equation, which I've listed here, but all of the components of this triangle, of the pyramid, can really play a role in helping to increase that selection response. So if we kind of break down the breeder's equation and think about how do we obtain genetic gain or increases in selection response, we can think about each component separately and how it relates to that pyramid. If we think about intensity, this really is kind of how do we increase the number of individuals that can be evaluated so we can actually select more individuals. And that can depend on your phenotyping strategies. If you're saving enough time, you can actually include more individuals in your populations, or the use of integrated software to be able to phenotype in an efficient manner. Accuracy is really defined as the degree of correlation between what you can estimate as your breeding value and what the true breeding value is, or what you can't actually see. And so the quality of your phenotype data and the quality of your data management and the links between those two are really going to play a role in how you estimate breeding values. Next is variance. So the ability to accurately estimate the biological component can really depend on how or which traits are actually measured. And so this involves data management and genotyping platforms to really link the phenotype to genotype, as well as phenomics. And then I think probably one of the most important parts of this equation is the generation time, because it has the biggest impact on gain. If you can decrease your generation interval, you can make the most gains. And so that really ties into where we're approaching advanced breeding, such as predictive breeding, in order to decrease the generation cycle and result in more selection, higher selection response. And again, that predictive breeding is really built on the foundation of all of these particular components. And so I think one thing to notice about this is phenomics plays a big role, data management. And then for this particular talk, we are going to focus on both of those, as well as touch a little bit about integrated software. So image-based phenotyping. The definition that I like to use is extracting the physical characteristics of an object through some type of imaging. And I say object because you can have different targets for what you're trying to phenotype. And these can span a broad scale. You can go from very small cellular phenotypes to tissue level, organ level, plant level, small population, or even just multiple populations across a given area. And so it can be used across all of these different scales. In addition to that, imaging can be defined in many different ways. If you think about the range of wavelengths that we've identified, what we actually see with our eyes is really limited to this visible spectrum. And so those are things we can capture with RGB cameras. But when we go outside of that is where we can really leverage the use of other types of sensors and obtain other types of imaging data. And so this may not be stuff we can see directly with our eyes, but we have tools that provide a way to develop something that we can see at some level. In addition to that, all of these different types of approaches and tools that span these wavelengths can be useful in a variety of different settings. Some of them can be very easy to use in the field or easy to use in the lab, but may not necessarily translate as well in other types of environments. And they can be used to identify different types of morphologies, as well as other physiological characteristics. And there's been quite a bit of research on all of these. But one thing to keep in mind as we think about expanding our horizon on what we can and can't see outside the visible spectrum, a lot of these approaches really result in a lot more data that can be generated, which increases the complexity of the analysis of what you get from these platforms. And so that's really where establishing a foundation where you can manage your data effectively really helps lead the way into using some of these other approaches. And so I'll transition a little bit to some of the things that I've learned throughout my time with BI OnRamp and how to create a successful imaging project. And so I'll spend a lot of time on the planning component. I think you'll find that an hour of planning will save many, many hours of stress and heartache down the road. And then I'll move into a very straightforward tutorial on how to use Fieldbook to capture images. And I'd like to start off by just kind of defining the focus of these particular components. We are going to be specifically talking about visible spectrum, so images that we obtain with an RGB camera, so the cameras that you find on your tablet or iPhone or DSLR type cameras, as well as using Fieldbook specifically for image capture and data management. And then really focused on more of an individual plant or organ level type target. And so planning, like the initial steps of trying to plan for an image-based phenotyping project is identifying what your target trait is. Spending a lot of time to determine what is the target component of your commodity that you're looking at, what is the trait that you're most interested in, and how to effectively capture that in an image. And so the first part of that is, can it actually be captured in an image? So a lot of geometric and color components, size, color counts, and shape are really easily captured in an image platform. And then you can use those later on to associate with other characteristics that may not be directly observed within your image. The second thing is to really think about how that particular trait is traditionally measured, uh, really evaluating what the benefits are of changing a given approach, and also trying to determine if there are ways that you can incorporate in your workflow to evaluate success. So comparing your image-based traits to manual measurements is a good way to kind of demonstrate with data that you're replicating that process in some form or fashion. Evaluating whether or not your imaging will require multiple images, depending on your target trait. So in the case of citrus, if you're trying to get an idea of color around the entire fruit, you may have to take multiple images, so of the front or the back, and that'll feed into how much time will be spent on imaging itself. And then really thinking about whether or not the image-based approach will be comparable to your manual approach. And a lot of this is focused on whether or not your traditional measurements are subjective and then moving into more of an objective measurement, or if you're going from qualitative or categorical measurements and actually quantifying it. I think this is really big when you think about color. And so if you can really think about these particular components, it'll help set the stage on how you evaluate and infer results from the image-based phenotypes that you'll collect. The next part is focusing on environment and methodology. So a lot of times when I go into a new imaging project, I spend a lot of time really looking at where the imaging is going to take place, as well as the resources that are available to capture those images. And this is what really impacts the design of an imaging setup. If we think about taking images in a lab, objects can often be brought to an imaging area, and so your platform can be stationary. You have a lot more control on lighting, so you can have a very stable light setup. And air conditioning has a huge impact on the quality of your images and the quantity you're able to obtain in any given unit of time. And so a lot of this can be kind of really a stationary setup where you have maybe some foam boards that block out most of the light and a tripod that you can mount your tablet on to take images. If we're thinking about a field setting, the imaging area may actually have to be brought to the target object. So focusing on a mobile type setup. You have a lot of variability in lighting that can be due to weather, time of day, or the size of the species. If you're imaging very big trees, you have to think about what approaches you would need to capture that. And so you need to really have a flexible solution for light control. You can have variability in condition, so rain and humidity. And if it's flowering season, you can anticipate everything being covered in a lot of pollen. We experience this quite a bit in maize. And again, air conditioning has a huge impact on quality and quantity. And here are some examples of some people in the lab who've been doing some imaging throughout the last year and a half or so. And then finally, if we think about methodology, this is really focused on how are you preparing your target object. So if you're imaging roots, you may have a series of steps that require root washing, or maybe you have to cut fruits, and that can be very messy. And that can really help decide whether or how durable your imaging platform needs to be. Consistency in who is imaging. So if you are using the same people to image everything, then you may not have to put as many resources into providing a foolproof setup design. So a designated area to put your target object, or a designated distance from your target object. However, I would err on the side of caution here and just always go with a foolproof setup design, and I'll talk about that a little bit later. Concurrent phenotyping. So a lot of this is while you're capturing images, are there other types of phenotyping going on in the lab or in the field? So is there juicing or grinding, or do you have to track units to compare different measurements outside of what you get from the image? And again, resources that are available. For example, if we have a situation where only one person is available and is doing all of the imaging and all of the prep work, I really like to think about how we can modify the setup and the workflow to save the most time and effort for a given project. And here are again some more examples. So when we think about planning, we have to think about what the components of a good image are so we can prepare our imaging area. And really, I think this kind of starts with an understanding, or at least a basic understanding, of what the image analysis process looks like. And so what I have are just a series of like general steps. There are many, many different types of algorithms and approaches out there that can do different types of corrections, but this is a basic workflow. And so you start out with an original image that you see here. We have some fruit in the contrasting background, a color card, and a size marker. And so we can take that image and use a particular area of the color card to do a white balance correction. You'll see it brightens up a little bit. It doesn't have as much of an impact on this image because lighting is pretty good. But in some cases, it can make a very big difference in the quality of your target objects. The next step would be some sort of thresholding, and this is based off color or different channels of the color spectrum. Here, we're really just thresholding on light against dark. And we can use that threshold to mask out the background of an image. And so we've applied a threshold here, and we can see that we have pretty good coverage of our fruit, but also some of the components of our color card. And so another step in masking is based off size. And so we can say, okay, well, I know a lot of my objects are so big, we can mask out all the objects that are below a certain size. And then what we end up with are objects that are ready to measure in some capacity, whether it's color or geometric measurements or whatnot. And so if we think about all of the steps in this particular analysis pipeline, we really need to focus on, okay, well, what can impact the quality of an image and whether it exists in the background or there's noise in the background that will really affect our ability to really be able to mask the fruit and provide some of these thresholds. And so I think to think about what impacts this particular pipeline is actually, instead of thinking about the components of a good image, we really need to think about what the components of a bad image are. And so shadows are the bane of my existence when it comes to imaging. They have a huge impact on the quality of your image and your ability to be able to measure what the target is in that. And so here's an example of a fruit that has quite a bit of shadowing. We may be able to identify the outline of this fruit, as you can see by the pink lines, but when it comes to thresholding and maybe extracting color values, we're going to be limited to a certain area of the fruit. And then there's going to be a lot of question on whether or not the measurements we're taking on color are actually accurate because of the effects of the dark shadows in the background. Additionally, shadows and other background interference, such as grid lines in this image, can also impact your ability to threshold. And it makes it very difficult to segment out your object from the background or even components of your object you may be interested in. So here's an example where in the second part of this image, you can see a lot of the grid lines are showing up after masking. And it's because there's still some contrast between those and the background. That's pretty equivalent to the fruit color itself. Next is object placement. And so if you're imaging multiple objects in a single image, really where you place those objects relative to each other can impact your ability to delineate objects within an image. So you can see that on the top left here, these two fruit are pretty close together and there's a little bit of shadowing in between. And so when we threshold and try to mask these, they actually get combined into a single object. And so we'll end up with phenotype data that is, instead of being for two oranges, we actually have one measurement, and we'll have to go back and manually correct this to get the measurements for 2 objects. And then some other components that are really important. If we look at this image, it seems to be pretty good. We have a contrasting background. We have our object of interest in the image, but we're really missing components that will allow us to do that color correction or that white balance, as well as an ability to convert the pixels of an image into an actual metric for size. And so how do we avoid the components of a bad image when we're doing an imaging project? A lot of it is focused on controlling the lighting to eliminate shadows. And again, this is the image I shared earlier. It can be as simple as maybe foam board around your imaging area to block out some of the light coming in from different angles. Also providing an additional source of light, such as the ring light in the top left, can help eliminate shadows in a lab setting. If you can't completely control lighting, really timing the imaging event during favorable conditions. So overcast outside, the time of day, or trying to reduce the amount of shadowing in some capacity. So if you're imaging smaller objects in the field, you could possibly use an umbrella over the object and your camera to reduce the impacts of direct sunlight. Being mindful of object placement and or providing guides for object placement. So this goes back to that foolproof method or foolproof setup. And so in the case of this particular tree here, you can see the base has different components, different 2x4s that are almost in a grid fashion. And so there's only one place where that pot can sit, fit snugly in this imaging platform. Additionally, you can have, in this case, notches and foam board. And so whoever's imaging knows that the fruit has to sit in any one of these given notches. If possible, you want to choose a background that provides maximum contrast, so dark on light, light on dark. And you can see that in this image with the notches, and you can also kind of see that here with the white and the darker fruit. You really want to try to avoid background noise. So that example with a gridded background, you want to try to have a blank background and avoid any of the other noise that could be present in any other type of gridded or actually I can't think of any other backgrounds, but that's a good example. You want to incorporate a color card in the image, and that provides you an opportunity to do white balance as well as advanced color corrections. And here's an example of a pretty cheap color card you can get from Amazon. It comes in a 2-pack for 13 bucks and it works pretty well. You can also buy more expensive ones if that's something you want to do. And finally, you want to incorporate a size marker in the image. And so that's what this little circular dot is here. It's 3/4 of an inch in diameter. And I can use that to say, okay, well, if this is 3/4 of an inch, how many pixels does it take to make 3/4? And I can translate that into the size of this particular fruit. And you want to do a lot of testing. Set up your platform, take some test images, look at how they look on your device, and adjust if needed. And again, all of these components are really targeted at increasing the accuracy within the breeder's equation. The higher quality phenotypes you're able to collect, the more accurate, the better chances you're going to increase that numerator and result in an increase in selection response. And so the next step is image acquisition and data management. So thinking about how do you plan on taking images? Are you going to use your phone, your tablet? Do you have a fancy DSLR camera? And then once you've decided on a device, how are you going to track your images? A lot of times these standard devices will append a very standard name to what your image is. It'll be like image 2140, 2141. And so how are you going to link them to the actual plot, plant, accession, treatment, replicate that that describe your target object? And then how are you going to link them to the actual trait that you're trying to capture in your image? And if you're trying to use something like Excel spreadsheets or things like that, you really have to think about if that method is going to negate any of the time savings you may gain from actually using imaging. And then thinking about data storage. If you are in the middle of an imaging project, you don't want to have to stop because you don't have enough space on your SD card. So just kind of looking up the capacity of your device and the size of the SD card you may need could help mitigate some of that stress of having to stop in the middle. Now, this is where I'm going to plug in Fieldbook because I think it's a really effective app at not only collecting other traits, but also for image acquisition and data management. And so Fieldbook is an Android-based app. It's open source, it's freely available on the Google Play Store. And it has a pretty simple setup. All you need is a field layout file. You can define some traits, and then you can actually collect data directly into your tablet. And so what you end up with, if we're thinking about images themselves, you end up with an image such as the one you see here. And on the bottom is the name of the image as it exists because we captured it in Fieldbook. And so what you can see here is that you actually get a unique ID, a trait ID, an image rep number. So if I took 2 images of this particular tree and trait, that number will be a sequence of those, and then a timestamp. And this is automatically appended to your images while you are collecting them in your app. And so if you go back later on to all of your data and try to figure out what is what, you're going to have all of that information tied directly to the image itself. Additionally, another plug here, Fieldbook has been undergoing quite a bit of improvement. And one part of that is focused on integrating external cameras into the app itself. And so through a grant funded by AG2PI, there really has been a focus on using external cameras because they can provide more rapid and controlled phenotyping. You can get higher quality images because your resolution will be much better. And they can be operated independently of the app. And some of the objectives are really to capture images from a set of external cameras within Fieldbook. And then another component, if you think about image quality and resolution, is really optimizing the file storage for these. And so that's actually pretty far along. Through communication with Trevor, they've targeted GoPro cameras, which are really nice because they are resilient to weather and things that you may experience in a breeding program. And they've successfully integrated that as part of a trait within Fieldbook. It's undergoing a lot of testing, but it's really close to being released and can be utilized by you guys. So keep an eye out on that update for Fieldbook and give it a go when it comes out. Okay, so now I'm going to take a step into more of the workshop component of this talk. And this is really focused on taking images with Fieldbook specifically. And I'll go through the series of steps needed to set up Fieldbook, take images, check some of your collection progress, and export those images so that they can be used for downstream use. And so this is really depends on, or it's based on the idea that you have downloaded Fieldbook on your device already. It's available on the Google Play Store. We can provide some links in chat if needed, but that is the first step, download Fieldbook. So once you download Fieldbook, your user interface, when you open the app, looks something like this. You have a list of 3 different things here: fields, traits, collect, export, settings, about. Very simple. I like that user interface. It's pretty straightforward. And so the first step is importing a field layout or an experiment layout. And so what you'll do is click this button here called Fields, and that opens up another window. And so we don't have anything loaded on this tablet yet. So what we need to do is click on this nice plus sign so we can import a field layout file that we've already created. So once we click that, we are able to navigate within the device's local storage to a folder that contains our field layout file. Here I've called it my imaging field layout file. And so what that file looks like, it's a CSV file with a plot ID in the first column. And so this is the unique ID across all of the accessions that we would be imaging. You have a plot number, an accession ID, and then you can have other information here, such as row and column information if you have a field gridded system. If you don't really have that particular setup, these are not required columns, and you can be pretty flexible in what you have included in this particular file. So once we select our file, this window will pop up. And this is where we define what our unique identifier is. In this case, it's plot ID. You can have another primary identifier. So as you're navigating through all of these individuals, this will be a kind of another layer of identification. In this case, we'll have an accession name, and then again, another layer of identification, plot number. And so we'll click import, and that field layout file is now in our tablet. So the next step would be creating a new trait. Since this is an imaging talk, we're going to create an imaging trait. So again, here's our home screen, and we'll select traits as the second option here. When we click on that button, I have video, so it's like not cooperating. We'll get this pop-up window here, and you end up with all of these different boxes here, and the first box is format, and so this is where you can choose the format of your trait. Fieldbook supports quite a bit of different formats, from numerical, categorical, all the way to GNSS, which are GPS coordinates. And so we're going to click on photo. You may see here that there's a USB camera option, and that's sort of the initial components of integrating external cameras into Fieldbook. We're going to use the internal camera of the tablet, and so we're just going to click the photo trait. Once we click that, we'll get this box popping up that we can name our traits. In this case, I've named it tree front, and you can provide some details that may guide whoever's collecting this data on what that trait actually means. So here I put north side of the tree, and then we'll select save. And once we do that, we can see that our trait now exists under our traits section. And then all of these other ones are default traits that get imported when you first download Fieldbook. We can toggle which traits we're interested in collecting at any given time. So here our tree front trait has a check mark in that box. So the next step is actually collecting data, and I'm going to focus this by using barcodes to navigate through our plots for this particular experiment. And so again, here's our home screen. The third step is collect. So we're going to click that. And what happens is, now that we have our field layout file and we have a trait created, we will get this, this description in this particular window. And so we can see that we have a plot ID. Here it's image project 1007. I've modified what displays here by clicking, I don't know if you can see my mouse, but you can see that I've displayed row as row 2 and column as 1, and you can actually customize this depending on what you want to see as you phenotype. In the middle part, you see tree front and north side of tree, so that's my trait and trait description. We have accession and plot number. And because this is an image trait, we have a nice little button here with a camera on it. And so if we want to collect a trait, and we know that we're already at that plot, we can click that camera button. But when you have larger experiments or projects, sometimes it gets a little crazy navigating what your target is at any given time. And so Fieldbook has the capability to scan barcodes and do navigation to your target automatically. And so there's this nice little button here with a barcode encompassed in a partial rectangle. We can click that and we can use barcodes that we put on our target objects. So if you see here is an orange that I put a QR code on. And then if I were to use my regular camera and read what's on that QR code, it's actually a unique plot ID. That's not for this experiment, but that's what is used to search your accessions and plot IDs within Fieldbook. So if we click on that barcode icon, this is a video kind of showing that. What will happen is we'll get our camera automatically access via Fieldbook, and this should loop through. And we can scan a barcode. On this case, I used a barcode on my display. It dings it and it navigates directly from 1007 to my plot 1001. So that's a nice little handy way to navigate through your plots.
And Heather, had you already taken a picture for 1001?
Yeah, I did. So you can see that an image was already taken there. So you'll see that and know that if you take another image, you're just going to have two, or whether or not you want to overwrite that image as well. And I'm trying to go to the next part, but there we go. Okay, so here's another video kind of showing what the image acquisition process looks like in Fieldbook. So I've already scanned a barcode, I've navigated to my plot. Now I want to take a picture. And this is a picture on my computer, but what happens when you click that camera button, it opens up the internal camera on your tablet. And so you'll see all the functionality of your camera here. So you can zoom in, zoom out, whatever other capabilities of your specific tablet. And you can take an image and it'll ask you if that image is okay, or if you want to retake it. If you click OK, that gets saved under your plot that you have navigated to. Okay, and then finally, what we end up with is an image for this particular trait and plot. And we can see that here, and we can go to the next plot by either scanning a barcode or navigating using these big black arrows. Videos complicate things a little bit. All right, so next, just a quick interlude of how to check your collection status and then exporting data. When you're in the process of collecting data, Fieldbook has an option to be able to check the status within a given field. And so there's this nice grid button icon here. And if you were to click that, it opens up kind of a visual of your field layout. I only have 12 plots here, but we can see that we took an image for 1001. So it has content for that particular trait. And we can use this as a guideline to determine whether or not we have missed anything or how close we are to completing imaging on that particular field. And then the final step is exporting data. So again, on this home screen, we have export. When you click that button, it provides an option on whether or not you want to export data locally to your tablet, or if you want to use the breeding API to do that. And I'm not going to cover that here, but if anybody has questions about that, we can talk about it another time, or try to maybe schedule some time to cover that in another seminar. But we're going to choose local storage. And once we click that, we'll get this particular window where we can choose the format of our output, whether it's a database file, which is really kind of another word for long format, a table file, which is your wide format. You can choose to export all of the columns in your field that you've imported, and all of the traits that exist in Fieldbook are only the ones that you've toggled a check mark next to. And then you can bundle all of the media data, which are your pictures, to that export file. And you end up with a zipped folder containing everything for that given layout and trait. So we're going to hit save. This window pops up and says, please cite me for using Fieldbook. So please do that. And then you can see at the bottom here that our file is being uploaded to our target folder. If we navigate to that folder, you can see we have a zipped file. If we unzip it, we get this folder called output. So here's our database file, here's our table file, and then all of the media associated with it. So our trait has its own media folder called tree front, and you can see our image that we've collected here with all of that information appended that I went over earlier. If I were to open this table, what's really nice is that our image name is now also in a table with all of the other info from our field, as well as the location of this particular media and the name of that image as well. And so you have kind of two ways to track this data. So with that, I'm going to kind of just go over some of the next steps and final thoughts. I'd like to mention that we've covered only kind of a small part of what image-based phenotyping encompasses here, but that small part really is a way to establish that the foundations needed for data management and doing some of the other more complex imaging approaches that are becoming readily available. And so we did an intro to the OnRamp initiative and imaging itself. I tried to provide a series of steps and guidelines for obtaining high quality images and really setting the stage for downstream data analyses and downstream use. The most logical next step is actually focused on how do we analyze these images, and that would be something all of its own. And I don't think I would have been able to include any of that here. But basically using this pipeline or customizing it to process your images. We have to keep in mind that, again, this is only a small part of what imaging actually encompasses. And so thinking about where we can go from here and what other types of approaches we can use. And I didn't have time to include some of the other advanced approaches such as machine learning, which can be used to do automated image processing. And so hopefully we can touch base on that at some point in this seminar series. So if you have a lot of background in that and want to give a talk, please reach out. And again, we haven't really covered what the step from image-based phenotyping to what we call high throughput phenotyping. And so really leveraging the technology that's available to increase the speed of data collection and move from maybe single plants to whole fields and beyond. So with that, I'd like to give thanks to a lot of people. I'm not going to go through these individually, but pretty much everybody here has contributed in some form or fashion to actual image acquisition, software development, our image analysis pipelines. So a lot of the images come from the Citrus group out of Fort Pierce. We also have a few that have come more recently from the Parlier program out in California. And so thank you all for all of your help with creating some of these images and helping me learn along the way on what really is the best components of a successful project. And I'd like to thank you guys for taking the time to participate in this seminar/workshop. And if you have any questions, I will take them now.